Hi, my name is Victoria Mullen, and this project is a literature review titled The Effects of Exosomes on Injured Tissue. Tissue repair is a highly coordinated, multi-step process that requires immune regulation, angiogenesis, extracellular matrix remodeling, and functional regeneration to occur in a precise sequence. When this coordination breaks down, healing can become impaired, leading to fibrosis or chronic tissue damage. Increasingly, research suggests that regeneration is driven less by direct cell replacement and more by paracrine signaling, meaning how cells communicate with each other. Exosomes, which are nanoscale extracellular vesicles carrying proteins, lipids, and nucleic acids, have emerged as key mediators of this type of communication. This review examines how exosomes influence tissue repair across injury models and what factors govern their regenerative potential. So the objective of this review was to evaluate current preclinical and translational evidence on the regenerative potential of exosomes in injured tissue. Specifically, we focused on their roles in intercellular communication, modulation of inflammation, activation of key reparative signaling pathways, and the impact of biomaterial-assisted delivery strategies on tissue-specific outcomes. For methods, we conducted a systemic search across PubMed, Scopus, Web of Science, and EBSCO host, including English language, in vitro, in vivo, preclinical, translational, and clinical studies. To be included, studies had to clearly define the injury model, exosome source, and delivery method, as well as the target tissue, and report quantifiable regenerative outcomes, including wound closure, angiogenesis, histological repair, or functional recovery. And what we found was that across models, exosomes derived from mesenchymal stromal cells, fibroblasts, macrophages, platelets, and endothelial cells consistently enhanced tissue repair in cutaneous, bone, neural, and ischemic injury settings. Reported outcomes included accelerated wound closure, increased vascularization, improved tissue architecture, and enhanced functional recovery. Importantly, delivery strategy emerged as a critical determinant of efficacy. So biomaterial-assisted platforms, including hydrogels, microneedle patches, and scaffolds, consistently improved exosome retention and sustained bioavailability, resulting in a more robust and reliable regenerative outcome compared to free exosome administration. So overall, exosomes function as integrative regulators of tissue repair. We found in our conclusion by coordinating immune modulation, angiogenesis, cellular survival, and regenerative signaling. And while the cargo of exosomes is important, our findings indicated that therapeutic success depends just as strongly on delivery design, including localization, timing, and interaction with the injury microenvironment. So future work should focus on standardizing exosome isolation, characterization, and dosing, as well as optimizing biomaterial-based delivery platforms and advancing controlled translational and clinical studies to evaluate the long-term safety and efficacy. And together with these standardizations, these advances will be essential for translating exosome-based therapies into reliable, clinically relevant regenerative treatments. Thank you.